Hi everyone, good evening. Can you hear me?
Yes, ma'am.
Okay. So, as we have generated the VCO file, and today we are going to perform our last step, that is the annotation. Okay, so for the variant annotation, we are using one online database, that is VP, which is Variant Effect Predictor. Okay, so I just want to show you. Yeah, is my screen is visible? So here we are going with the VP ensemble. VP, that stands for Variant Effect Predictor. So this VP actually it determines the effect of your variant, like whether it is a SNP or whether it is a indel, means like insertion, deletion, or copy number variation, or it is like on a gene, on a transcript, or any protein sequence as well as any regulatory region. So that we are going to find it out with the help of VEP. Okay, just give me a 1 minute. Okay, so what we have to do is we need to give the simply input of our coordinates, means like the VCF file where the nucleotide changed, and it is finding out like whether your gene and transcript affected by any variant or not. Then it will find it out the location of the variants, like where it is located, whether it is in a upstream region, downstream region, or any regulatory region, or it is in a coding sequence or in a non-coding sequence. So that we are going to find it out with the help of like VEP. Consequences also, which is like whether it is a missense variant, stop gain variant, or it's a frame shift variant. So that also we can able to find out with the help of VEP. Then whether it is a known variant or a novel variant, means like whether it is already present in any one of the database or it's a novel variant. So that information also you will be able to get in the VEP. Then SIFT and polyphase score, like these are the pathogenicity prediction score, means it will tell you the information about the pathogens, like whether it is changing your protein function or structure. So this is what the information we are going to find it out with the help of uh, VEP, variant effect predictor. So once you open the VEP, after that what you have to do is we just have to open the web interface here. Yeah. Now here it is showing you the human uh, 38 assembly. But maybe if you're having any another species, you just have to go with the change species and you can select whatever the species you're checking about. Then here you need to be upload your VCF file. So you just have to choose the VCF file from the CRISPR folder, which is your GATK VCF file, GATK output. Open it. So the file is loaded. Now here it is asking you from which database you want the information. There are four databases like Ensemble GenCode, Ensemble GenCode Basic Transcript, RefSeq, and Ensemble GenCode and RefSeq. So as RefSeq is giving you the validated information, so we are going with the RefSeq, but the choice is yours, completely yours, that which database you need to select. Then here you are having the additional configurations. So additional configurations means what, like what columns you have in your result table. So what columns you want in your result table. So if you will see here in the identifiers, so basically now identifiers like gene symbol and the transcript is selected. But maybe if you want to visualize any another column in your result table, you can just check that box and it will appear. So same thing you can check in each and every section and you can select the columns which are which you want to appear in a result table. But by default, whatever the columns are selected, for that, using that column also, you will get enough information. So no need to check any extra. But still, again, the choice is yours, and you just have to click on run. So it is in a queued. We just have to wait till it is run. Everyone needs to perform. I hope you are performing. Yeah, it's running. Done. So here it is showing you all the like annotation is done. You just have to click on the view result. So in the view result, first of all, it is showing you the statistics, like a summary statistics. How many variants are processed? Total 53. How many variants are filtered out? Zero. How many are novel variant and how many are already existing variant? You can see out of 53, 38 are novel variant and 15 are known variant. Then how many are from overlap genes? How many are from transcripts? Then how many are from regulatory features? So that also you're getting. Then here you are getting the consequences, like how many are frame shift variant, how many are missense variant, are protein altering variant. So that percentage you are getting. But as most of the time we are interested with the coding sequences, so in a coding region, how we are having the variant, like in a coding sequences, how much percentage we are having about the frame shift or protein altering variant, because protein altering variant, it can be harmful. But with this table, we are not able to understand anything basically. So that's why like with this statistics and like this table and the image, we are not able to understand anything. We need the information like what is a gene, then it is a protein coding or not, or what is a reference allele, what is a altered allele. So that's why here the table is given over here. And this table you have to download in a text format. Like there is options we are having here, VCF, VEP and text. So if you will download it into the text format, we can better visualize it with the help of some columns. So in the downloads, you can see that this VEP file is downloaded. You can just cut this file, put it into the CRISPR folder, and you can rename it as VEP output. Now this VEP output we are opening in Excel. You just have to select all files, delimited it, tab separated, and finish. So once you finish it, here it is showing you all the columns. Now I'm just going to save as this file in an Excel workbook format so that you can visualize the column-wise data properly. So Excel workbook, yeah. Now here you can see it is showing you the uploaded variations, right? Means like there are different columns we are having. So I will explain you one by one. So basically this uploaded variation, this is a column which contains the identifiers, but in a VCF file also, if you remember, we don't have any identifiers there. So that's why, just a second. So basically, hmm. So here these are the uploaded variations where we don't have any identifier, so that's empty. Then you're having the location. So this location means like where the variation is located. So here we are having the 17 means what? This is your chromosome number that on 17th chromosome it is located from this base position to this base position. Means like it is giving the format like chromosome colon start position hyphen end position of the base. Then this is your altered allele. What is your allele present in the read? That is your altered allele it is given. Then here it is giving the consequences. So there are consequences means what? Basically, this will tell you what type of variant it is. Means like this columns is describe the predicted functional consequences of the variant on affected gene or a transcripts. Means it is showing you like it is a missense, frame shift or a nonsense variant. So if you just want to see what other options we are having here, uh, I will show you by applying the filter. I'm just selecting any one of the value for now, going with the data and apply the filter. So if you will see that you're having the frame shift, intergenic, intron, so let us consider you don't want anything, you just want the protein coding variant, protein altering variant, because these are the harmful. So you can just uncheck all and you can only select about the protein altering variant and say okay. So it is going to show you all those variants which are protein altering, right? So this is how you can apply the filter. Then impact it is showing. So this is telling you the impact of the data, like impact of the variant, whether it is a highly impacted or whether it is having a low impact or moderate or modifier. There are four options coming under the impact: high, low, moderate, and modifier. So high means the impact of the variant is high. Moderate means uh, somewhat it is showing the impact. Modifier means we cannot say anything for sure. And low means like it is a low impacted variant. So maybe if you only want the high impacted variant, you can just select the and you can apply the filter. Then symbol. So from which gene you're having this variant, that it is showing to you. So there are different genes we are having. You can see as per the data, we are interested with the BRCA1 variant. So you can see the BRCA1 is there. You can apply the filter. So it is showing you the variant for the BRCA1 gene only, right? Sorry. Hmm. Then as this is showing you the gene name, this is a gene ID. Okay, this is a symbol and this is the ID. Then feature, in what feature we are having this variant means like it's showing you the feature type. So the feature type actually it specifies the type of genomic feature where the variant is located, such as transcript or any regulatory region or any intergenic region. You can see regulatory feature or a transcript. Transcript means nothing but what? RNA, like a product of transcription. And if it is a transcript, so ID of the transcript it is showing you. Then biotype, like what type of variant, what at what type of variant it is, like it is a protein coding or it is in a LNC RNA means like a long non-coding RNA or it is in a promoter region where it is located. So most of the times we are interested with the protein coding variant. So you can apply the filter that protein coding variant if you want. Okay. Then exon. So it is showing you the exon numbers basically that how many exons are there and out of that how many are mutated. So you can see here you are getting the 14 by 23. It means in this transcript there are 23 exons are there, but out of that 14 are mutated. Okay. There are 23 exons are there, but out of that 14 are mutated. So this is what the exon and same about the intron. So actually by default it is showing you the format, how they are representing it one by one. But Excel is automatically taking the format as a date, 1 January. But actually it's a one by one, which means like there are total one exon is there, but out of that one is mutated. Here also, if you will see 1 March, means actually they just want to represent it as a 1 by 3. Means there are total 3 exons, but out of that, 1 is mutated. Okay? I hope it is clear to everyone. Any doubt up to now? Yeah, I will come up to you, Joydeep sir, because I need to see your screen for that. So let me just complete this part and then I will come up to you. Okay? Hmm. Okay, Daimat sir, I will share this VCF file after the session. Then next we are having this HGVS. HGVS is nothing but what? It's a Hugo Gene Variation Society. So according to that variation society, if you have a name, C, it is showing like HGVSC and HGVSP. So HGVSC, that is for the coding sequence, and P, that is for the protein. So if you have any information according to those variation societies for the coding sequence or for the protein, so that you will be able to see over here. But you can see these columns are empty because we don't have any information about it. Then cDNA position. So that the position of variation or position of that variant at the cDNA level, then at the CDS level, then at the protein level, and at the amino acid level. So that position of variant as per the different features, you will get it here. Because like if it's a protein coding, so obviously in the protein sequence, at which position you're having that variant, at which amino acid you're having that variant, you need to find it out. Then here you're having the codons information also. You can see here, I will show you. That previously, there was like this many amino acids, but later it was just altered as S. Means what some deletion happens over here, and what kind of deletion happens, so that at the codon level also it is giving. So that codon level mention over here. Then existing variant. So this existing variant, it will tell you that whether your variant is a known or a novel. So if you're having any ID in the particular row, like right now we are having the RSID. Do you know RSID is a prefix of which database? Do you know? NCBI? No. NCBI or like in a NCBI, which database? Like, no, it's a DBSNP database ID. RS. If you're having any ID with the prefix RS, so it's a DBSNP database ID. Okay. So here you are having the variant from the DBSNP database. But for those variants which are not having any information about the IDs, those are the novel variants. For that, you're not getting any information from any database. Oh, all right. Then here reference allele, like your allele in the reference, then what uploaded allele. Then here you are having the distance. So this is the distance from the TSS transcriptional start site. Then here you are having the strand information. The variant is located on which strand. So if you're having a -1, it means it is located on reverse strand, and 1, like which is a +1, that is a forward strand. Then flags, actually these are some statistical score which you don't need to be worried about. Then here you are having the symbol, like symbol means what? Symbol source actually, like from which source they have taken the gene symbol. So entries. Then HGNC, which is a Hugo Gene Nomenclature Committee. There is one committee which is giving a name to gene. So if that ID is present, then this main selection, these are some annotation database. This TSL, APRIS, these are some statistical value we don't need to be worried about. Actually, now here, these two columns are the most important column for us, which is a SIFT and polyphane score. So right now we don't have any value because maybe the data is not changing any protein structure as well as function. The variant are not changing any protein function as well as structure. But these values are the pathogenicity prediction scores. Means uh, if I will show you about the SIFT and polyphane score, yeah. So if you are going with the pathogenicity prediction score, it is showing you the values for the SIFT and polyphene. So what the SIFT values represent, it's predict like whether the amino acid substitution is likely to be affect the protein function or not. Means what? It will tell you that whether this amino acid change it is affecting your protein function or not. Means whether it is changing your protein function or not. Because if protein function is changed, it means this is harmful, right? So there are two values, there are two possibilities we are having for the SIP score. Like either it is a deleterious or a tolerated. Deleterious means what? It is harmful, and tolerated means it is not harmful. So if the value of a SIFT score is less than 0.05, it is deleterious. It is going to be damaged for sure. And if it's greater than or equals to 0.05, it can be tolerated. So this is how you will be able to get the SIFT score. Right now in our data we don't have that value, but in future if you are getting, so you can just filter according to this cutoff. And next we are having that is a polyphane score. So basically polyphane score, it is predicting the effect of amino acid substitution on structure and function. Means SIFT is telling you about the function, but polyphane it will tell you the structure as well as function. So if the polyphane score is greater than 0.908, it is probably damaging, means it is going to be damaged. If it is in this cutoff, it is possibly damaging, it can be damaged. Then if it is less than 0.446 is benign, which is like not tumorous. And if the value is not known, you will get the unknown. So this is how you're having the SIFT and polyphane score. And rest of the columns like clinical significance, it is telling you whether it is a pathogenic, benign and all. So if you have any PubMed articles or any motif information, so this is how you are getting all the information about your variant over here. And how you want your variant, uh, sorry, how you want your variant, accordingly you can just apply the filters and you can select the important variants. Okay. So this is about the variant annotation. So here your CRISPR is completed. Now, whoever is having any doubt, you can tell me. First doubt is a Joydeep sir, like tell me what is the problem? Just show me the uh, show me your error, like what error you're facing.
Hello, ma'am. Am I
Yes,
audible?
sir.
Okay, ma'am. I am facing problem of that whenever it is generating the BAM file. That
Yeah, that's what.
the
Like, can you share your screen because I need to see the command once.
okay, ma'am. I'm sharing.
Yeah, yeah.
Is it visible, ma'am?
Yeah.
Okay. So Here is the command, ma'am. I just provided.
Uh-huh. Uh, but paired reads have different names. Can you do the LS, sir, please? No, just let me see the. It is showing you can see paired reads have different names. Can you just try it with the normal FASTQ file instead of trim file? Because I think there is some error in the trimming. Can you just try it with the without trimming? This SRR files.
Okay, okay, ma'am.
Yeah. Yashaswini ma'am, about the certification, just put the message or your query in the WhatsApp group. As I'm the trainer, so I don't have any idea about it that much.
Ma'am, I have one question.
Yes,
Um, so we are performing the germline mutation in here.
right.
But why not the somatic mutation?
It just the choice is yours because I told you the algorithm for the somatic as well, right? Mutate to two. It's just like instead of haplotype caller, you need to change that name. And if you are interested with the somatic variations, because germline variations they are transferring from one generation to the another, right? So that's why we are interested right now in a germline, but not
Uh,
necessary. You can call the mutate to two as well.
but ma'am, we are just processing for the BRCA one, but it is expressing some tissues which are not in germline. So that's why that's my
Yeah,
question.
yeah, I can because it's not like that on a chromosome 17 because we are aligning the reads to the 17th chromosome, but it's not like that on a 17th chromosome we are only having the BRCA one, right?
Yes, ma'am.
Yeah, there are other features also. So yeah, obviously that also maybe if there is a variation, you will get the information about it. But that's what I've told you to apply the filters for whatever the information you're interested about, right?
Yeah,
So I think it is processing now.
yes, ma'am. I think.
Yeah, because maybe it is having any error. It will stop up to this point. But I think your trimming is having some issue, sir. So just solve the trimming problem properly.
Okay, ma'am.
Hmm. Yeah, for now you can just go with this and you can proceed further because this is just for demo. But
Okay.
after everything is done, like just check once the trimming.
Ma'am,
Uh,
can you tell us any visualization tool like IGB? So how we can
yes.
visualize it?
Yes, I can tell you about the IGV. Okay, I will tell you quickly. Just let me share the screen. You can stop the screen sharing.
Okay, ma'am.
And Amit sir, actually, fourth lecture is also uploaded. I think link is already shared. But if not, I will just convey to the management. Maybe they will share the link. Okay? Because I have uploaded the sessions today only. So, no, Sneha ma'am, you don't need to run this command. Actually, these are some extra command in my script, which I was using previously. This is a variation filtration. We don't need to perform any variation filtration in a CRISPR. This is for the DNA-seq, so no need to run this command. Whatever the commands I have told you, they are the important, like these are the important commands only. Yeah, that's what, like Amit sir, I will just convey the message. I will recheck it. Okay, you will get all the recordings. Don't worry. Okay, so if you want to visualize the variant, so there is one tool we are having called IGV. You can download it and install it in the Windows as well. No need for Linux. So here it is a uh, visualization, like this is a GUI based tool which is showing you the, yeah, which is showing you the GUI. And here this is what a tool. Basically, this panel is for your variant, like this is for your chromosomes. Then this middle panel where you can visualize your variants, and the last panel is your reference panel. So basically, for the variation visualization, uh, you require some files. So I'm just creating one folder in the CRISPR by the name of IGV, and I'm just giving all the files which are required. So the first file, because IGV just for the visualization, it is not doing anything by itself. Whatever the input files we are providing, it is using that files only. So I'm using a BAM file, then chromosome, that is your reference genome, and the GATK output VCF file. I'm going to copy it and paste it into the IGV folder. Okay, so once these files are there in IGV folder, actually IGV required the indexing of GATK output. Sorry, IGV requires the indexing of your reference genome as well as the BAM file. Okay, so what you have to do, you have to go with the tools, run IGV tools, and here you have to select the option index, and then you have to browse it. So I'm just selecting the folder where I'm having the file. So first I'm loading the chromosome 17, that is the reference genome. Open it, and then you just have to run. So it is showing you done message. Now you have to browse again the BAM file. So select the BAM file, open, and click on run. So it is showing you done. So indexing is done. You can see in the folder some BAI, that is the indexed files are created. BAM index and FASTA index. Now you have to upload the genome. So go with the genome, then load genome from file, and again you have to select the folder here. So as this is what your reference genome is loaded, now you have to load your BAM file. So go with the file, load from file, and select the BAM file, not the indexed files. Okay, open it. So here the BAM file is there. Now you have to load the VCF file, GATK output, VCF, and open. So here, whatever the pics we are getting, these are the variants. But if you want to visualize a variant for a specific location, so let us consider, I want to visualize the variants for a BRCA1 gene. So I'll go with the symbol, apply the filter for the BRCA1, and I will select any one of the position for now. I will select this position. This position, either you can give the name of gene or a position in a VP format and click on go. So, okay, we don't have any variation over here for the visualization. Just a second. I will take some another location. Actually, they are not having that much depth. That's why we are not able to visualize the reads over there. But maybe if they are having a depth more than 10, so at that time you will get the reads over here and you can visualize what variant is present in the reference and what present in the reads there. So this is how the process actually, but the data they are not having the depth properly and that's why we are not able to visualize it. But still, just a minute, I will check for others. Okay, so yeah, we don't have that much depth actually, but this is what the process you can follow for the visualization. Okay, yeah. Same we need to plus. Yes, yes, Nikita ma'am. If you are going to deal with any another organism or as well, just input files will be different, output files will be different, commands will be same. Okay, and the process will be. You're welcome, sir. Yeah, is there any doubt to anyone else? Actually, IDV requires that indexing because use of indexing to increase the speed, and that's why IGV require those files inside the folder. Okay, Sneha ma'am? Okay, yeah. Any another question? Uh, I'm sharing the attendance link as well. Just give me a 1 minute. In the meantime, if you have any question, please ask me.
Hello, ma'am.
Yeah, hi.
Ma'am, suppose if we are using induced pluripotent stem cells, we can use the same way or not.
Whatever the cells you're using, ma'am, or whatever the organism, the steps will be same for variation calling or for the uh, like CRISPR analysis.
Okay.
Yeah.
Suppose if one stem cells differentiate into a renewal cells and a differentiated cell, so how we can control the procedure, like how we can. Hello.
Uh, Actually, sorry, I don't have any idea about it. Means like these are some biological part, and I'm an analyst, so
Okay, ma'am.
yeah. Because my work is like once I will have a FASTQ file, I can show you how to perform, but
Okay, ma'am. I will try. I will contact you if it is required.
yes, okay.
Thank you.
For actual sequencing, how can further the control and treated sequence? Ms. Nikita ma'am, I didn't get your question. Can you just elaborate it? I have shared the attendance link as well. Please mark the attendance. For gene expression analysis, no, this is not the same process. For the gene expression analysis, actually, it's coming under the RNA-seq. So there we have to compare the differential expression. So obviously tools will be different and the procedure, the analysis, everything will be different. So that's what like you need to learn the RNA-seq analysis for that for the differential expression. Okay? Yeah. There is someone in the group who is asking for WSL installation. I don't know what is her name. So, like, just tell me if you join the session so that I can guide you how to install. She's asking continuously about the commands and all. Mm, okay, all right then. So if you don't have any doubt, we can disconnect. This is our CRISPR session. About the certificate, you will get a message in the WhatsApp group. As about the certification, it is coming under the management team. So, okay, so here your CRISPR analysis is completed. I hope whatever the things we discuss, it is clear to everyone. Still, if you have some problem, you can just put your messages in the WhatsApp group. But before that, I will just suggest to go through the recording and perform it once again so that you will get more confidence about it. Okay? All right then. Uh, Attendance link. Okay, I will share the attendance link again. Yeah. Uh, yeah. It's opening now? Can you check? Uh, Actually, these are some basic commands what we have discussed, but there are some alternative tools also. So you can check what are the alternative tools we are having, and you can try with that. So for the trimming, actually there is a cut adapt, Trimomatic tool is there. Cut adapt and Trimomatic. Then for the variation calling analysis, instead of GATK, you can use the BCF tools as well. BCF tools. So you can try with that. You will get the manual online, how to install, how to proceed. So yeah. Is there any question? Attendance link I have shared 2 times, sir, already. Okay, I will share it again. Yeah. Is it opening now in your system? Okay. Then what can I do, sir? Means like I'm sharing this third time right now. Just try it once now. Yeah, so I think Amit sir, you're having some network issue. Please check that. Copy paste it somewhere so that you can maybe whenever your network will work, it will open. Okay? Yeah. Okay, so all right then. Bye. Have a great day.